I came across an article that I thought would be interesting. 3D nucleosome mapping opens up new visions of dynamic DNA architecture by David Kopage on January 16th and 19th, excuse me, of 2026. Let me just show it to you here. You can find it here at Discovery Institute's Science and Culture. And if you just type in 4D nucleosome mapping opens up new visions of dynamic DNA architecture, you'll definitely find it. I was going to just go through this article with you, and we're going to talk about it, the big points, the takeaways, and worldview aspects of it. So let's jump into it. What was this article about? What is this article about? It's about the 2D nucleosome. What is that? Okay. Well, a 2D nucleosome describes a three-dimensional organization of DNA inside the nucleus, plus how the organization changes over time. It moves beyond just pure DNA as a simple code, like a text, like linear code, but it focuses on the folding, looping, and positioning and motion as a functional, meaningful feature. So it's more than just text. It's more than just a simple line of code. The 3D aspect of it has impact on what's going on as well. And that doesn't count like epigenetics, but that's besides the point. A major scientific milestone, excuse me, the researchers produced the most comprehensive map yet of the human genome architecture in space and time, integrated multiple experimental methods. This work represents a accumulation of years of collab research into how DNA is physically organized within the cell. And if you can imagine, this would be quite an undertaking. The findings, key big findings that we should think about and know about. Each cell contains tens of thousands of DNA looping interactions. Yeah, tens of thousands. And this is each cell, by the way. Bringing distant genetic regions into functional contact with each other. So they'll think of like a spaghetti, which I think we'll talk about a little later. It's kind of like spaghetti just looping around each other. That's kind of what's happening here. And they're brought in close contact with each other. Genome structure varies between cell type and even between individual cells despite the identical DNA sequence. So this might be one of the reasons why we see different cellular cells doing different things, like having different functions. Large-scale architectural features, loops, domains, and compartments play a major role in gene regulation. Why does structural matter? Well, before we get to that, I just want to note, please like, subscribe, comment down below if you like this content. If you want to hear more about it, definitely subscribe because I'd love to have you on board and be part of this. Why structure matters. A DNA function depends not only on sequence, but also on shape and spatial arrangement. Genes are often regulated by elements located far along the DNA strand, but positioned nearby in 3D space. The genome behaves like a static instruction manual and more like the genome behaves less like a static instruction manual and more like a responsive spatial organization system. There's a lot going on in the cell, and I think this has great implications here, which we'll talk about a little later here. Right now, genome architecture changes during development, cell division, stress, and differentiation. These structural changes help explain how one genome can produce many different cell types. Disruptions to folding or spatial organization can lead to disease, even if the gene sequence remains intact. So genetically, you might be totally fine, but because of disruptions in folding and spatial organization and 3D structure, you could have disease that manifests itself, which is interesting. Mechanisms of organization. Spatial proteins organize DNA into functional neighborhoods that limit or enable interactions. Molecular clustering and compartmentalization concentrate regulatory machinery where it is needed. Organization occurs in multiple scales, from local loops to whole chromosome territories. There's a hierarchical design to this all. DNA is organized in a nested layers. We have nucleosomes and chromatin fibers, and we have loops, domains, and nuclear components. These layers work together to regulate accessibility, timing, coordination of gene activity. So if you just have one of these things out of order, you could have issues, which we talked about just a few minutes ago. If you want to know, if you want to download my quick guide, Why Science Points to the Creator, please check it out down below in the description. All right, the big picture. The genome is dynamic, structured, and context dependent, not random or mechanically simple. It's very complex, integrated. Understanding genome architecture reshapes how scientists think about genetics development and disease. And this is super important because the more we understand, hopefully we can solve some of our big disease issues in today's day and age. Worldview perspectives that we really want to look at, it does really undermine the reductionist DNA linear code model that so many of us just assume to be the case. It shows that a function depends on a multi-level organization, timing, and context, and highlights coordination, hierarchy, and integration, not just mere chemical determinism. And it weakens the simplistic gene alone explains everything narrative. I think this is important because it shows that there's more to life than just a simple code, which makes life way more complex, way more meaningful, and way more meaning packed within this 3D organization of DNA and other codes that tell the cell what to do and how to do it. If you like this video, please like, subscribe, comment down below. Don't want you to miss any videos in the future. Also, please pick up the quick guide, Why Science Points to Your Creator. You can find that in the description below. See you next time.